Good morning, good afternoon. As the industry started already to test their potential risk product, we start developing method and testing complex nitrosamine, meaning API-related nitrosamine, and we clearly see that this is now the main challenge of this testing step. My presentation is focused on this complex nitrosamine and their formation during drug product manufacturing. So what are our options to remediate formation of complex nitrosamine in the drug product? We have mainly three options presented here, going from the lower time and cost consuming to the higher time and cost consuming. The first option is to set an appropriate acceptable intake. After alignment with health authority based on scientific proposal, this is the simplest and fastest option to be compliant. The second option is to reduce the risk by using low nitrite excipient. At the present, we are focusing on excipient with high load that will obviously have the highest impact if they contain nitrite. We will come back to this option in a specific slide. The third option, but the less preferable, is a reformulation of the product to avoid use of identified materials containing nitrosating entity or work on the manufacturing process. We will discuss further in the last slide. So why is it not preferable? The reformulation is time consuming since we will need to have new development with several PKs, manufacturing of batches, 6 month stability and submission. Roughly, it will take few years until the product will be marketed again. This activity has also an important cost and will impact development capacity of the companies. Therefore, profitability of the product will need to be evaluated if the reformulation path has to be initiated. So what are our limitations for setting an acceptable intake? Acceptable intake proposal is based on read-across, proposing a surrogate or in vivo or in vivo studies, and sometimes based on available literature. The challenge here is that there is no clear guidance on how to do a read-across, but also on how to de-risk complex nitrosamine. So if the actual tools are found not acceptable, then it is urgent to develop new tools and to support research to guide the industry. The risk benefit to patient, this impossibility to be able to de-risk a complex nitrosamine impact new launch of product, but also impact development of new products due to the testing burden in developing these complex analytical methods. No remediation work can be initiated due to uncertainty of the limit. So we are recommending to set interim limits based on actual data available and ICH-M7 principle until further guidance on studies to be performed for nitrosamine will be available. For the excipient role in remediation of complex nitrosamine, there is a recent initiative from Lhasa to collect nitrite analytical results from the industry. Main excipient suppliers are aware of the need to reduce levels of nitrite, but not necessarily ready to remediate because of their level of knowledge or because large investment will be needed, and sometimes also because the pharma industry is not their main market. Here also we need to support research for helping the supplier to remediate. Some suppliers started to remediate, but availability of these grades will take time and consequently possibility to remediate the drug product is delayed. Sometimes the source of nitrite is inherent to the excipient. Also, most of suppliers refuse to have a limit of nitrite in their COA, meaning that it will be challenging to put in place a control strategy in the finished product. So the question here is how we can remediate complex nitrosamine for a finished product if excipient suppliers are not ready with their remediation. There are several aspects to consider for remediation via reformulation, and we are only at the beginning of the research. First, the API. The industry has to better understand the impact of the physical properties of the API, For example, crystallinity of the API or the impact of the micronization on complex nitrosamine formation during finished product manufacturing. Parameters of the finished product manufacturing might need to be better understood, such as wet process parameters, temperature of drying, loss of drying, or the pH. Can we use scavengers? We have seen in the past strong impact of certain scavengers in the pK of some product. Also, we are limited by the IID levels in the quantities to be used to see an effect. In general, the solid-solid reaction has to be better understood if it is from the kinetic perspective or the parameters that impact the reactivity of the API. It looks like that at this level of understanding, reformulation is applicable to new product development, but not for commercial product. Thank you for your attention.